Welcome to Cell Biology for Pharmacy. This video is the second in a series on the cell cycle. In this video, you will receive an overview of the eukaryotic cell cycle. Let us begin. In general, cells reproduce by duplicating their contents and dividing into two cells. The most basic function of the cell cycle is to duplicate accurately the vast amounts of DNA in the chromosomes and then segregate the duplicated DNA into two genetically identical daughter cells. Now, the key aspect of the cell cycle here is the accuracy of the duplication in order to produce two cells that have the exact same DNA. As you can imagine, when a cell divides into two daughter cells, each of the daughter cells will have nearly exactly half of the mass of the parent cell. In order to prevent the cells becoming smaller and smaller as they divide again and again, cells have to grow double in size before they undergo cell division. Therefore, another important aspect of the cell cycle is that the processes that govern cell growth must coordinate with those that govern cell division. The duration of the cell cycle vary among different types of cells and cells of different organisms. In this table, you can see several examples of cell cycle times of different cell types. As you can see, cell cycle times vary from several minutes to multiple hours. For example, frog embryo cells divide once every 30 minutes, while mammalian fibroblasts divide once every 20 hours. The cell cycle of eukaryotic cells usually consists of four main phases, namely the M phase, where cell division occurs, the G1 phase, the S phase, and the G2 phase in that order. In the M phase, two processes occur, which are mitosis and cytokinesis. We will discuss in detail each of these phases in this video. However, under the microscope, the two main observable events of the cell cycle are when the nucleus divides and the genetic materials are segregated, as you can see on the left, which is what mitosis is, and also when the cell divides into two new cells, which is known as cytokinesis. Both of these events are part of the M phase of the cell cycle. Now, the period between one M phase and the next is called the interphase. So all of this is called the interphase. Deceptively, if you look under the microscope and peek at a cell, which is at the interphase, you won't see much of interest. All that you may be able to observe is that the cell is increasing in size. However, the interphase is a very busy time for a cell. It consists of the S phase, in which all of the cell's DNA is duplicated, flanked by the G1 and G2 phases, during which the cell continues to grow. Now, in these gap phases, the G1 and G2 phases are called gap phases. The cell continuously monitors its internal and external environments. By monitoring the external environment, the cell ensures that the conditions needed for reproduction are just right. And by monitoring its internal environment, namely the contents of the cell itself, it ensures that all preparations are complete before the S phase begins, in the case of G1, or before the M phase begins, in the case of G2. Later, we'll see what happens to cells that undergo the S phase or the M phase before the necessary preparations are completed. So, in summary, the cell cycle consists of two large phases, the M phase and the interphase. The M phase is what's called the mitosis, during which cell division occurs, and also cytokinesis occurs in the M phase. And the interphase consists of the G1 phase, in which the cell grows and all other normal processes occur, the S phase, in which all genetic material of the cell is duplicated, and the G2 phase, which is similar to that of the G1, where the cell continues to grow and prepares for mitosis. An important function of the cell cycle is to determine whether the cell is ready to continue to the next phase. So, at particular points in G1 and G2, the cell decides whether to continue to the next phase or pause to allow more time for the cell to grow or complete its preparations. As the M phase is the only time the cell is visibly changing when observed through a microscope, it's important to study what exactly happens during this period. The M phase consists of mitosis, which is the separation of the nucleus and the genetic material, and cytokinesis, which is the actual physical division of the cell. As you can see here, mitosis consists of 5 subphases, which we will discuss in detail in a future session. In summary, mitosis consists of the prophase, the prometaphase, the metaphase, the anaphase, and the telophase, which is then followed by cytokinesis. During the interphase, cells generally continue to perform their normal metabolic processes, such as transcribing genes, synthesizing proteins, and growing in mass. This provides the time needed for the cell to duplicate all of its cytoplasmic organelles, all of the components of the cell, which will be shared between the 2 daughter cells created after cell division. Most cells spend most of their time in interphase, growing in size, transcribing genes, and synthesizing proteins. However, in some organisms and cell types, the G1 and G2 phases are shortened. For example, in frog embryos, the first cell divisions that occur are very quick in order to subdivide the large egg cell into as many smaller cells as possible. Because the G1 and G2 phases are shortened, the cells don't have enough time to grow before being divided again, resulting in smaller and smaller cells. That's it for this video. In the next video, we'll dive deeper into how the cell can control the cell cycle and why it's important for healthy growth of the cell. Thank you.